Hello, everyone, and happy Wednesday. We're going to go ahead and get started here. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix, and our webcast today will be given by Gabe Rudy, who is our Vice President of Product and Engineering, and Gabe will present the use of VarSeq as a clinical NGS platform. Gabe, it's all yours.
All right. Thanks, Cheryl. So thanks for joining us today. I'm very excited to actually share with you some of the things that we've been working on at Golden Helix to add to VarSeq and now is in VarSeq that is very applicable to this very exciting field of using next-generation sequencing in clinical tests. So to start with, just a little bit of housekeeping. We will, as usual, have some time at the end to take questions. And if you're thinking about something as I'm explaining or demonstrating something, you don't need to write it down on a scrap of paper. Just go ahead and use this questions pane of your little GoToMeeting interface. It can kind of be expanded there on the right. And with that in there, at the end, I can actually read them, and Cheryl will moderate as we go through and answer those questions. If we don't get to everything, don't fret. We will follow up and make sure every question is answered to the best of our abilities. So with that, we can start the fun here. So what I'd like to cover in this webcast is about the clinical context that next-generation sequencing is being used, and specifically what are sort of the bioinformatic requirements that necessitate solutions that can handle all of these things, such as capturing the workflow of a test and the lab, capturing the knowledge that people working with that test are essentially the insights that they're coming up with as they're doing their work, and the decisions that they're making, and also handling the fact that some tests are still quite expansive in their search space, and you have lots of candidates to look for in terms of candidate variants, candidate genes that might make sense in a diagnostic setting, and how to use both filtering and ranking approaches to efficiently use the time of the folks involved performing this work. So if you're not familiar with us or with this product VarSeq that we'll be talking about today, Golden Helix has a long history in the genomics, bioinformatics, and research support community here. We've been probably one of the few companies in this space that has our history span into the past millennium, actually. And that history has gone through a lot of technological revolutions and changes and disruptions as we've seen things go from genotype focus to now with sequencing and NGS sequencing variant focused work. And in 2003, we actually announced that we were looking to leverage a lot of our mature technology and build out a tool focused on this problem of variant annotation, filtering, and interpretation. And with that announcement, we started talking to a lot of our existing users and folks that we've been in contact with to essentially refine and define this product. And we're very excited to launch it last October. And then in the 6 months since then, there's two main things we've been focusing on. And the first is essentially seeing the mass swell of interest in using this in a clinical context and people onboarding VarSeq into their process. We were very keen to work with those groups and understand their requirements. And this is essentially our development process in any context, but we really spent the last 6 months building out our features and to support these requirements. And a lot of what I'll be sharing you today will be things that have been newly added since that initial launch. And if you look at our release notes, which are in our manual, and you can find through our public documentation, you'll see also how incredibly responsive we are to all of our existing users and incrementally improving every single component of the software. It's just been a blast to see the uptake and the context that people are using VarSeq, and the team here is excited to be improving it into all of those for all of those folks. So this is just a sampling of some of the groups that were gracious enough with their time to talk with us and really iterate on what we're be talking about today, these requirements for using this in a clinical and repeatable workflow context. And this is really what makes it all possible. So what I'm talking about is really an aggregate of this expertise, not individual expertise on my part, but we are trying to represent what I think is the commonalities between labs. So every lab is unique, but there's a lot that is shared in terms of what is needed from this portion of the workflow. So when I say this portion of the workflow, let's step back a bit and talk about next generation sequencing being used in the clinical context. So the main vehicle for this progression, and it really has taken off over the past couple of years, is what's called LDTs, or laboratory developed tests. This is something that the FDA realizes that they can't keep up with the pace of innovation and have every individual test need be satisfied with off-the-shelf pre-validated tests. So this is the context in which labs can encode their own expertise into a test that satisfies the needs of the physicians or institution that they are serving, and they have to then be responsible for their own validation and within some context be able to then show that they are effectively producing tests that can then go on to get reimbursed and work just like any other assay. The thing that has really popularized this recently is the fact that labs don't have to start from scratch. There's a lot of support from vendors and from existing precedents to be able to very quickly set up the in-house expertise to run these tests. And what that looks like is essentially acquiring the proper equipment. So you need something like a desktop sequencer and often machines to do library preparation, including your target selection, amplification, and some of these machine-specific work to bind and adapt the DNA to go onto the flow cells, or in the case of Ion Torrent, the individual chips. But all this is, and even the informatics up to the point of, running alignment, variant calling, demultiplexing, is going to be pre-baked into your vendor solutions. You can certainly stray from that if it makes sense. You have specialized work that needs specialized informatics there. But all in all, this type of workflow can be done without too much focus on what the application is. A lot of these steps are going to be generic, whether you're running a single gene NGS panel or a 17 gene. You might change your load levels and how much you multiplex with all that. But again, even these types of choices, you can start with some vendor provided solutions. If you're doing hereditary cancer risk, there are targeted panels out there for BRCA1 and 2. I believe there are vendor-provided cardiomyopathy. And if not, you're definitely not trailblazers in going into one of these wider ranges of looking for sort of classes of symptoms being covered by, I know GeneDx has like a 17-gene neuropathy panel. There's definitely Noonan syndrome panels, epilepsy panels, etc. So these may be the focus of your lab. You pick one of these to get going, and certainly there's a lot of support with off-the-shelf hardware and kits to get to the point where you have your variants called, but then that's where the validation and the uniqueness of each individual test and lab need to be well vetted and standardized so that you can repeat the steps that you need to do to get to a result. And that step includes also the final work of what level of interpretation you are deeming appropriate. So whether you're just simply calling variants that are well classified and having someone sign off on their presence or absence and supporting their quality, or you're looking at potential novel variants and interpreting those and capturing that knowledge. This can also be the place that is essentially the make or break for whether this test can be economical. A lot of these steps are going to be standard laboratory procedures, but here is where if you don't have the right interpretation workflows set up, you could spend hours per sample when economically it makes sense for a given, and I've heard this, that for say a well-characterized small gene panel, they are a lab that I've been talking to says that their target is to get it down to, say, 5 minutes per sample and have their workflow support that level of throughput, so that they can make sense in terms of how they bill out the test and how much of the total lab resources it requires to run that test. So let's talk more about then what I mean by this interpretation workflow. What are the things that need to be captured here? So this is a screenshot of VarSeq, and you could imagine this as the project that the person developing the assay, essentially validating with, say, R&D samples or samples that have known mutations in them at specific levels so that you can tune your sensitivity and specificity. This is sort of the environment that they're going to be using, and what's great about it is everything that they choose to do here in terms of setting the thresholds. Again, this is going to be unique to the test. What are the read depths you expect to have? This is an oncogene panel, so you expect to have very high read depths, and you're looking at essentially the mutation frequency of the tumor versus the normal, and you may expect that to be within a fixed 1 to 30% range, and you may expect that you are not going to be doing sort of novel variant interpretation. You want to stick to variants that have been cataloged in the cosmic cancer database. So these types of things you may set up, and then you also are doing things like deciding which specific versions of these annotations and other algorithms that are run here. Even things like which columns are visible, what the order is, and the tables that you have set up. Here's a view of these variants at a variant by variant level, and here's a table view that actually groups them by gene, so you can kind of look gene by gene to see what you're looking for. This is the details view. For every variant, it pulls out all the information. So all of these are in fact captured in what we're calling a workflow here. And so this includes the choices that you go through on import. So those get defined and saved with a workflow. Again, the things we've added is annotations, the individual parameters used in filters, and all of these views. The way VarSeq does that is that project that you just saw can be saved as a template. We call them project templates, and then a new project can be created using that template and will reproduce precisely every single step that was taken to produce that project, and also reproduce the essentially the dashboard or the workspace in terms of how you are viewing and setting up your view into that data. So this includes things like, if you chose to annotate against Cosmic 71, that template will have that locked down. Cosmic 72 may be available, but you don't want to revalidate all of your workflows until the NM that you decide to do that. And so every single step, as well as every single parameter and choice of the specific versions, are kept inside of that template. And with our new version, we've also added this log view, which keeps the same type of information incrementally added as time-stamped with the user, the action, and the details. So in this case, you can see I modified a filter, disabled it. I added a database in which one, we'll talk about that feature in the future. But that also allows you to sort of support the regulatory environment that is often seen in a clinical setting. Okay, so let's go ahead and run through VarSeq. So this may be, in some cases, the first demonstration of VarSeq, but I'm also going to assume that we're going to just mostly focus on the things we just talked about, reproducing a workflow with a new set of samples, capturing the log, etc. And to demonstrate that, I'm going to sort of work in the context of someone who was developing their own tests. And this is a project, let's say, of someone developing their gene panel assay for a sort of standard hotspot cancer panel. It has 14,000 individual variants. We filtered down to those that are called as high quality by the variant caller. We used a read depth filter here. Again, we're looking at things that are in cosmic. And then for this sample, you can see I can switch which sample I'm looking at here. You can see the variants changing for which variants are present here. And for each variant, we can look at its annotations. In this case, I have a sort of embedded web tab here that when I click on, say, this MET variant. I can look at its cosmic information, which tissues it is. So this looks like a colon or stomach cancer oriented mutation, etc. So let's go ahead and do one more customization to this project. We're going to add in one more annotation, and then let's say we're happy with this and we're going to rerun it and rerun it as we would when we sort of decide that we're saving a template and then going into production. In this case, let's go ahead and we notice we still had 12 variants left after all these filters, and most likely what's going on is we haven't really accounted for some of these variants being in the germline. So to do that, I'm going to add in these, again, all these annotations are available in our public repository. When you use them, they get downloaded. They can be quite small, like this 112 megabyte one. But this is the mutations that have been detected in 17 individuals that are over 110 years old. So these are all their germline mutations. And so clearly, you know, if you've lived that long, you're not likely to be harboring driver cancer mutations. So we can use that as an annotation source, and we can use that as a filter. When I add that, very quickly it adds this group of columns here. And again, everything in this, this choice of how I display data is kept as part of my project. So I can say I want to hide maybe this and just focus on looking at this frequency information. Every column you can click on to get documentation about the how this was created and curated, a little histogram of the data. And in our case, we wanted to actually use this as a filter. So I can right-click on it, say create a filter, and now it adds this to my list of existing filters. And let's say we're looking for, you know, we expect our interesting cancer mutations to not be in this data set, so there's 5 that are not present, or maybe they are extremely rare. Whether that makes sense or not, I'm not sure, but let's sort of keep that as rare or missing. So now I have changed my final filtering results for the sample down to 5 variants. I'm happy with that, and I'm happy with my project. It's in a good spot for exporting to Excel, which might be the way I deliver this data onto, say, a colleague or back to, we'll talk about more about how to generate reports and capture other information from here. But this may be all we need to start with. I like this setup. Let's go ahead and then save this, as I mentioned, as a template. Again, what this is not the data in this project, but simply the information about how this project was constructed. And we'll call this sort of our, maybe this is our cancer gene panel V1. And so you might version these, and as you decide to rerun your validation, come back, update to new annotations or algorithms, etc., and have a V2. Maybe you have a different design in your panel, etc. as well. So let's go ahead and save our V1 template. And I'm not going to save this project, but I'm just going to create a new project using that template. So there it is, my cancer gene panel V1. I clearly could have put a lot more descriptive information about my choices of things there. But let's say now I have a batch of data coming off of my MySeq, and so this is a batch for, you know, batch 1 maybe coming off, and I'm ready to import those samples using this template. So now I'm in production mode. I finished doing the R&D for the sample, or for this assay. Oh, I already had something called that. Let's go ahead and call it batch 2. So we warn you if you're about to create a project that already exists. Okay, so now this is my batch 2 samples. I'm going to go ahead and grab, let's say, these 5 samples here. So it could be different samples than the ones we were doing the R&D on. And again, all of the settings that were used when I configured this template are saved. I can choose to maybe set up some controls. Maybe there's no controls in this individual run. Let's go ahead and hit finish. And very quickly, actually, the entire project has been reconstructed. And so now I'm looking at just these samples, and again, going from sample to sample, you can see which genes are present and can pull up their cosmic mutations and look at their tissues, and again, all these samples look like they're coming from a some sort of stomach or colorectal cancer. All right, so I hope that's very clear in terms of how easy it is to use VarSeq as your encoding of that lab and test-specific knowledge, your thresholds, and in fact, any of these things that we've added as annotations here could have come from your own internal databases, capturing some of the expertise or historical log of data that is relevant to the test that you are constructing. And so let's go back to our agenda and talk about knowledge capture. So again, what we've done so far is more or less just run in an automated way the type of annotations we're doing. Maybe a couple variants come out, maybe we export those to text files or Excel files, and you can export them in various different contexts. And it captures actually all of those rich hyperlinks, like those ones from Cosmic. Those can be in your Excel file as well. But maybe you also want to have more of a place to generate a report, and instead of having to copy and paste this out into a Word document, etc., we've embedded in our new version the ability to construct what we call project notes. So these are you can actually create more than one per project, and so you can kind of construct one that's at sort of a batch level, sort of logging the steps you're doing. This could be thought of as your lab notebook, or it could be thought of as a place to actually construct per sample detailed evidence information that can be pulled out, and you can pull it out as an HTML or PDF file. It's a complete rich text editor experience. And most importantly, you're in VarSeq, it gets saved with your project, and it can pull information from any of these views that we have in VarSeq. The details view, which drills down on the currently selected variant, the table itself, you could just copy out a whole row, and the genome browse view, which we'll demonstrate as well. A lot of rich context of what's going on in that genomic location. The second thing we've added is, okay, so that's a great way to have sort of free-form note-taking. But what about the work that you might do when interpreting a single variant? And there's a lot of different contexts and workflows people have shared with us that they would like to support. And this is a flexible tool that can really be adapted to pretty much any way in which you want to catalog variants outside of a given project so that they're global. Like you do some work, it's not sort of locked inside of that project, but also very flexible to adapt to how you set up your workflow. So what happens is as you select a variant, we place its identification information up here. It's a variant position and ref alt. And then these fields here get populated with anything that's in your database, and you can have multiple databases. We've set up one here based on, say, a neuropathy panel. And again, these are actually very customizable as well. You can set up classification levels, add your own. We have a couple templates, like such as the ACMG variant classification that you could start with and customize from there. And anytime you work in this context and do some work and type in some stuff, you hit save, and it gets added to that database, the exact precise information from your project and your assessment, and is also never lost. So if you go ahead and change this classification at a later date, you'll see who changed it when and what it was previously, and in fact, you can also revert to previous information, previous assessments. But you can also do more you can do, besides just sort of cataloging things, you can also do things like keep track of which samples you've seen it before, or have one user enter in their information and then another user open up the project. Let's say the lab director comes in and adds their sign off, and then you can filter off to variants that have been signed off and use that to generate a report. So it can be very amendable to different workflows. Certainly there's no pre-baked tool that's going to be flexible to do that, and we need something like this that is very, very flexible. One thing to keep in mind is we also make this very easy to set up. You can just start with having a single file on a network drive or a local drive that is your entire database, and that will work. If you want to allow for potential high concurrency or the robustness of having a full relational database, you can talk to existing Postgres or MySQL instances to host all of this data as well. All right, so those are some really cool features, but you really don't get a sense of them until we see them in context. So let's go ahead and run a project where we can get to work with these new abilities to capture the knowledge insights you're having as you're running your tests. So we did a cancer gene panel. Next, let's do a hereditary gene panel. And this is actually opening this project. I'll first walk through a bit of the filters, since we have a filter tab here, and we'll come back to this notes pane. but this filter tab shows the filters that we applied. So standard filters on redeath genotypes. In this case, we're looking for very rare variants that are potential missense or loss of function in their protein changing effect. And we're using rareness as defined by 1% or not present novel in the 6,500 exomes. And recently, the Broad Institute released what they call the EXACT project, the Exome Aggregate Consortium, which has over 60,000 exomes, and we're using the latest version of that. We just released 0.3's curated version of that data set, so we're using that as an annotation source. And so now we have, based on this filter, this sample here, you can have multiple samples in your project, just as we saw before, and you can certainly go back and forth between them. But you can actually click on each variant here, and I actually have my genome browse view showing this sample's variant information, including its coverage, where it is in the gene. You can get a lot of genomic context in this. So let's say we're using this new notes feature. So again, you can hit plus, you can add a note, as well as many of these other things. One thing I actually forgot to demonstrate was this log view, which is also in this just by adding a separate view tab. What that looks like is any annotation that's been applied. So here's applying that exact thing. We have the exact version with the specific version, and when it was curated and all the details there, as well as things like when I rename or change a filter, all of those are captured. And so you do have everything you would need to support sort of a regulatory framework where you need to capture specific actions. So back to our notes view here. Remember, we can actually grab information from various contexts. So if I'm looking at, say, a details view of this variant, I can copy and paste, information here, and that's what I did up here. I grabbed the genotype information. You can also, when looking at a genome browse view, you can insert a genome browse bookmark and capture and call it whatever you want. And this is actually Genome Browse bookmark. What that looks like is when you click on it, it jumps to that specific view. Let's say I want to zoom out and capture a little context of where this variant is in the gene, and also add in, you can customize how you want your Genome Browse views tracks to contain. So I can add in any of these sources. These are often sources we would annotate against. Here's the latest April version of ClinVar. Can also be visualized, and can do things like customize its label. And I want to show its clinical significance. So sure enough, this is a variant here that is known as, or classified as pathogenic by ClinVar. And let's go ahead and, this is some great context here. We have information that gives you clear confidence in the presence of the variant. It's right in the middle of this amplicon. You can kind of see that stair-step amplicon capture perspective. We're right in the middle of an exon of this gene, what gene it's in. So we can go ahead and insert a screenshot from Genome Browse right into our notes, right? So there it is, captures all that information, and of course you can type anything else you would like here. One thing I'd also like to demonstrate, we can copy things from the details pane, the Genome Browse view, and the tables. So I have a separate table view here that's just really focused on the individual fields that I want to critically capture in, say, a copy and paste action. So I can copy this entire row as an HTML table and paste it in here, or just hit Control-V to paste it in here. And this captures it plus all those hyperlinks. So let's go ahead and keep this and export this as a HTML file. You can also export it as a PDF, which would be great for sort of dumping into, say, a LIMS system, etc. And when I export that to a PDF or HTML, you'll remember the last path you saved it as. I saved it in my documents, so let me just open that up for you guys here. So there it is, right? It's got the images, it's got the hyperlinks. These hyperlinks are live, so I can click on this and go to ClinVar and see this, this existing pathogenic mutation classification for this disease that's pathogenic, and look at the supporting evidence, the PubMed literature for it, etc. All right, so that's the note capturing ability. And one other thing I mentioned was the ability to have these variant databases. So let's open up a tab here. And again, you could have this already pre-set up in your template, your project, etc. In this case, you can create a new database, and as I mentioned, that can be backed by a single file or a full-blown relational database instance. All your previously accessed ones are here in the order that you previously used them. So this is a neuropathy gene panel, and I created a little database for this neuropathy gene panel. And so this is what it will look like for a variant you've never seen before. Each one of these has its own little tooltip. So this is the ACMG classification. And again, it's really easy to customize this. If I went in here, I could actually very quickly change or what this means. If this was this is not a sign off, but this is like the GR sign off or something like that. If I have a very specific person, I wanted to say this was for. Easy to customize this. Your data gets preserved. You don't lose data by simply adding fields and changing field types, etc., and tweaking the documentation. But generally, you're going to get this set up the way you want it and then leave it. So now, let's say I look at this variant, this pathogenic variant, and it shows up in my database before. Yesterday, I added it as an annotation. You can see which sample I saw it in. Maybe I want to update its classification to now be just likely pathogenic, and I say new evidence of presence in unaffected as my reason for that. Go ahead and hit save. Shows up now in my list of previous assessments. And again, it's easy to actually revert to a previous assessment. And you can again have someone come in and say, someone else logs in and they're the director and they can sign off on this. And these are easy things to filter on. And what I mean by that is, although this is looking like sort of a form-based view, and you can use your mouse or your keyboard to sort of go up and down and look at each variant, you can also use it as a way of you can actually grab all of these fields, which phenotype, classification, and notes, etc., and add them as columns to your spreadsheet, just like you would add any other annotation source. So let's do that real quick. So annotate with data source. So we have the standard user annotations that we've downloaded, and now we have this new variant database section. And here's that gene panel data source that I was just modifying. So I can go ahead and select that, and it will add it here as a new set of columns. We can create this convenient Boolean column that just says, is it in or out of here? And here's our recent annotations, likely pathogenic, GR did sign off on this, etc. And these can be things that you can create filters on. And so, your workflow may have a version of that filters down on only the ones that have been signed off on, so that they're easy to sort of export out to an Excel file, etc. So very flexible in terms of how you use this. The point is, once you set it up, you have all the capabilities of VarSeq to make it part of your standard practice, your standard workflow, with very little concern for having, say, human error coming up by manually following a bunch of steps. All right, so back to our agenda. Covered knowledge capture. Now finally prioritizing which variants to look at, which genes to look at, etc. This is probably not going to come up as much if you're doing, say, a really small, narrow gene panel, like, say, a hereditary cancer panel, including BRCA1 and 2, or a carrier screening panel with CFTR, etc. But if you're in more of a very large panel, potentially with many phenotypes represented, and you have something you know about the sample that you're looking at, or very commonly you're doing exome analysis on trios or quads. We support various different configurations there. You're going to want to think about, how do I narrow the potential search space here? And certainly you can do this from a filtering perspective, but you can also choose to allow filtering to go up to a certain point and then use a ranking approach that leverages what you know about the sample and what we know about in our sort of biomedical databases, the relationship between patients' known phenotypes that you might see in a differential diagnosis provided by a physician, and genes, because there's a lot of literature out there that captures all of that. So in this case, I have a trio. It's been filtered with sort of standard rare disease model, looking for variants that are either in a recessive compound het or homozygous state, or de novo dominant, potentially an X-linked if you're male. And in this context, it's looking it's not aggressively filtering down to say only genes that are in OMIM, et cetera, because that would certainly narrow things down. But it is providing some level of filtering, which we get to about 600 variants. So at this point, we might want to switch over to a ranking approach. And with our latest version of VarSeq, we've incorporated FORank, which is an algorithm that does provide phenotype-based gene ranking. And it runs on your entire data set. So no matter, and you can start at the very top and just look at the highly ranked genes for every single variant. But certainly that's going to have some noise because there's going to be a lot of variants, that are intronic or synonymous and not all that interesting, very common, etc. So you can choose sort of at what point you lean on a ranking-based approach and what point you do some cleanup on the filtering size and really harmonize these two concepts. So what it's actually going to do is prompt you, this algorithm, to enter terms that represent sort of these differential diagnosis type assessments of the phenotype using the human phenotype ontology terms. There's about 16,000. This is a great biomedical database that's very current and certainly if you and we have a sort of a very convenient way to enter terms in here, certainly it's going to be easy to find a term representing pretty much anything that you would be able to capture. And what the algorithm does is using those terms as a starting point, it traverses these two biomedical databases of the HPO and gene ontology GO databases and essentially looks for the shortest connection between genes and these terms where multiple terms leading to a gene can also sort of emphasize its gene score. And then we provide a rank version of that score, and we also provide a path back from that gene to the shortest path back to the terms that you entered. And it's much better to see what this all means when you actually are entering in this into sort of a more real-life scenario. So let's go ahead and switch back into VarSeq to demonstrate this. All right, so back into VarSeq, I'm going to open up this project, which has had something that looks very close to our standard trio templates. So we provide some starter templates for doing things like trio analysis. They are inherently very conservative. We're not trying to filter down to nothing. And so what it looks like in this case is we have 13 variants. This is actually from that screenshot, the same data that follow a de novo mutation pattern. Certainly I would expect some of those to be false positives purely because you don't expect that many true de novo events within coding regions per generation. But here are about 500 or so variants that are compound het, recessive homozygous. In this case, the sample is, the patient is female, so we're going to ignore and disable. You can just go up here and say disable or enable this specific chain, and we're also not looking at a dominant model. Both parents are unaffected. But in this case, we can also, this is something we've added in our recent our templates, is just have a way of saying, well, are there any variants in ClinVar that have a known pathogenic classification that are extremely rare and, say, present in a heterozygous state in the child? And this can be just a sidebar, but very useful one to potentially find some variants there that would fall maybe outside of some of these other filters. Certainly, you kind of expect these to show up over here as well. So in this case, I have 600 variants, and in our gene-based view of this data, so on the left is the genes. As you click on a gene, you get to see the variants in that gene. We have it grouped to 244 genes. We have information about whether that gene is in a compound het state. You can actually hide some of this information if we want to make this a bit more compact. And we also have a gene list here applied, whether this is one of the ACMD incidental finding genes, such as APOB. About 50 genes there that have been suggested to report on variants of a certain level of severity. And now we're going to add this new algorithm we have called FORank. So these are its outputs, documentation in here about the algorithm. It's modeled heavily on the Fever algorithm by Mark Yandel's group. It's our own implementation. And let's go ahead and run this. It needs to know what the gene names are that it's going to use for its gene ranking, and then it needs to know what our phenotype terms will be. And these can be entered for each sample in the project. If you have multiple trios, you can certainly enter different terms for each sample. The output is sample specific. In this case, our one affected child here is the one we're going to work with. And so I have, in this case, a list of terms provided by the physician. They have their colloquial names. So this case is sort of a global developmental delay, skeletal muscle issues. They noted particularly having a protruding jaw. So using the form of that, we would go here is, yep, that's the protruding jaw term, as well as generally they discovered this, at birth, this is a sort of pediatric case, hearing loss, so let's look for something related to hearing. So obviously there's going to be terms that specialize these general terms into very specific versions, and there'll be sort of general things like hearing impairment. Let's see if I can find something that captures the fact that this happened at birth. So here, let's do this, congenital conductive hearing impairment. And we also want to go with, okay, they did mention the spine being flattened, so using the platyspondal. Wait, where am I at? Platyspond. Yeah, that. And also, generally, there was a, yeah, let's go with global developmental delay, right? So again, you don't have to be so perfect in these terms because these are all going to be related, of course. If you put in a general term, it's going to be able to connect to the specific term if there was an association with a specific term, and the other way around. So certainly being able to capture some of these things, you might go very narrow, you might go very wide. In this case, let's just use that as our phenotype capture for this sample. And now we have these new columns showing up at our gene level. And this path is going to be a great way of just looking very simply at these 200 genes and saying, oh, which of these terms did I enter is really most closely associated with this. So our gene rank is going to be something that we're going to sort on. And by doing that, it will give us the ranking approach on top of what we have as our existing filter approach. And then as we look at then the genes, and certainly, it's not like you expect every time the first gene to be the most relevant, but this gene rank score ranges from 0 to 1, and things that are sort of below the 60th or 70th percentile are by and large are going to be things that you would be suspicious of them being related to the diagnosis that you're trying to achieve here. All right, so here we have our results. We have, let's go ahead and sort by this, right? And here is global developmental delay coming up with very high relevance, and some of the flattened spine and that protruding jaw phenotype is coming up a number of times. And so as you click on an individual gene, you can get to its variants that are in that gene. I can sort of focus on that variant level view. I actually have information about the sample. Okay, so it's heterozygous here, and then its mother and its father are next. They're both referenced, so this is coming from our de novo filtering process. And as I click on the variant itself, it's probably better to look at this in this view. It's very easy to go down. It's a novel variant. It's in SMAD4. I can pull up the information about its CDOT and PDOT, the way I would report this variant. Lots of link outs to its OMIM and other types of gene level information. And interestingly enough, it is in ClinVar, and it has Meyer syndrome. So we may not know what that is directly. We can pull up a view here. We can also pull up a genome browse view to focus in on this variant just to validate its evidence. So I actually have the 3 BAM files for this child, mother, father present, and you can see the read depth support is very good. So this is the type of view I might dump into a screenshot for a project note, etc. And you can see, there's essentially no support for this variant allele of a C. So the fact that it's a de novo looks very self-evident. Let's go back to our ClinVar link. We can pop this up. You can keep this as a separate window. You can also just sort of dump it into one of your, this is an embedded genome or web view here, and pathogenic and information about the symptoms. This might start to make sense. You can copy, and if you want to like get out of using the web view here, you can jump on out and open up a web browser and zoom in on one of these links as well. I just copied out that link. So here is one of these great NCBI resources cataloging phenotypes with a whole bunch of descriptive information about that phenotype, and sure enough, there's that protruding jaw that the gene ontology information linked to this gene from one of our inputs. So in this case, this de novo mutation was the one that we were looking for, and it happened to be the first ranked in this list, but certainly just being able to look at the fact that the top 10 genes here might be of more interest, and then it starts to very quickly drop out, and you've gone down from having to look at 200 genes to reasonably having to look at, say, 10 genes or so. And with more aggressive filtering, you might even be able to narrow that even further. All right, so that about wraps up those three topics, and in summary, I hope I've been able to show the way in which we built VarSeq to support defining these workflows that capture sort of the uniqueness of each test, the unique workflows of each lab, really allowing that to be the place that they capture the efficiency and value add that the specialization of a lab brings to the laboratory developed test process, and the repeatability of that to reduce potential for human error and simply to support the throughput that is necessary to make these cost-effective tools or cost-effective tests. So, one other thing that we really focus on is scaling. So VarSeq, we worked with exomes, we worked with the gene panels. You can even go up into whole genomes. We don't see that often in say, a laboratory sense, but we have researchers doing that on more of a exploratory context. And the other thing that really supports this environment, this context of using VarSeq, is our simple licensing model. A lot of vendors in the space want to sort of tack on to the per sample throughput that you're doing in a lab and add some cost that's upfront and some cost that's per sample. We really don't find that's conducive to the way people want to have certainty about their financial planning and being able to upfront be able to account for getting the tools they need in place, and then not have to worry about the gray areas of things like, well, what if I needed to rerun a sample because it failed? Am I paying another click for that sample going through this process? Or how about all those times up on the R&D phase where I'm running over and over again many different test samples to really nail down those sensitivity and specificity? If you look at getting VarSeq for 3 or 4 users in your group, some on the tech side, some on the medical director side, etc. We have a very aggressive bundling pricing that just sets you up to have a yearly model for that, and a lot of certainty that it will be there and supported for your lab as it scales, even as it does very well and runs very many samples. All right, so with that, we could always point you to the Golden Helix website. We take a lot of care to put all of the material that we could produce, such as this webcast up there, and it will be up there in short order. Previous webcasts, ebooks that Dr. Andreas Scher has published on this topic of clinical testing as well as other topics. So many great things to look at there. You can request evaluations as well of VarSeq, etc. And with that, we will hand it over to Cheryl, who will fill you in on the latest from Golden Helix, and I will start grokking some of these questions, and happy to take as many as we have time for.
Thank you so much, Gabe. As Gabe mentioned, you are free to enter your questions into that question pane. Our next webcast will be coming up on May 13th, and that will be presented by Greta Lindsay-Peterson, who's our Director of Product Management and Quality. Invitations will be coming out in a couple of weeks, so look for those. And also, as Gabe already mentioned, our webcast recording and slides will be up hopefully by the end of the week, if not today. That's all for me, so I will dig in on some questions. Let's see here. Should dbSNP filtering be done beforehand, or does VarSeq have that built in?
Right, so we don't do any filtering beforehand. We have the ability to start with a completely empty project where all you have is the variant data that has been merged from your multiple samples. And we have the ability to start with what we call our starter templates, which include a couple filters on common things. And dbSNP IDs can be things that you see from your incoming variant files. There's an identifier field that may have those, and we can hyperlink those and follow those hyperlinks to dbSNP. And that one has been moved, but here's another dbSNP link. You can also add the latest version of dbSNP, so we keep up to date with those as well. You'll see them in our public annotation repository. We have dbSNP 142, and adding that as an annotation source allows you to be able to do things like create filters on that. In fact, let me do that really quick. I believe I have dbSNP 142 downloaded. Yeah, there it is. So if I add that, notice this workflow starts by looking at 50,000 variants. So when we do annotations like this, we're actually getting annotations for those 50,000, even if we're only narrowing our current view down to 600. So it takes 20 or 30 seconds or so to run this against the 100 million variants in dbSNP. But with this type of annotation, you can see it over here on the right, we also have a field created called like in dbSNP. This would be a nice simple Boolean field that you can use as a filter. Clicking on this, you can see, for example, of these 600 existing variants that I filtered to 500, 770 of those, about 93%. Are in dbSNP, some of them are not. I can add that as a filter just like anything else. So that's that question.
This next one's in two parts, so I will start with the first part and then move on to the second part. Can you access the underlying data, example publications, for a variant that is classified as pathogenic?
Yep, yeah, of course. So let's go back to our SMAD4 gene variant here. So in the details pane, we saw there was information about this being in ClinVar. One thing to note is ClinVar records are pairings between individual mutations and diseases. So one classification may be relevant for Meyer syndrome, another classification might be relevant for a different disease name. In this case, they had sort of a not provided disease name and a 0-star rating for that, but they have a record for it. Now, ClinVar has a great link out to various information. Let me sort of make this bigger here about on PubMed, etc. So you can go and see citations from the submitters about this variant in PubMed and pull up the individual mutation information there. That's just one source. You can certainly actually say go to the OMIM page for SMAD4. And this will have plenty of other citations on the bottom. There's about 40 of them about the gene. HGNC is also one of my favorite links for gene level aggregate information. They link out to mouse model databases, and as well as a lot of these great things like gene cards, Reactome, so if you're looking for pathway level information. So certainly it's really great to have all of these hyperlinks and annotations in here because they can be a starting point for that exploratory process. And you can actually, let's say you really wanted to save some of these things that you've captured here, you can actually create like new web views and have this one be the OMIM page and then that one be the HGNC page, and your project will sort of keep all this. It's like keeping the tabs up in your browser when you did a specific project. It's nice to come back and have all those at your fingertips.
Can HGMD be added as an annotation source with the appropriate HGMD license?
Yes, the answer is yes. With the appropriate HGMD license, of course, is the disclaimer there. So the license we would need is the one in which you can get a text version of the HGMD available. You wouldn't send that to us since we don't have that license, but we have helped other users and other groups take that data and get it into a form that's appropriate for annotation. And that's very simple. We have the ability to convert pretty much any standard genomic file, so whether that's a bed file, a GTF file, a VCF file, or just tab delimited or comma delimited data, we can convert those into annotation sources that you apply in your workflows that are private, and so those can be part of your templates as well. And for HGMD, there's certainly a couple things you want to get right to make sure indels are represented canonically, etc. If you reach out to us, we can work with you to run some scripts to prepare that data. It's actually relatively straightforward. We've done it before. In fact, I have, let's see, I have a little sample of some of the edge cases here. One of these. Well, maybe not, maybe it's on my other computer, my development machine. But anyway, yes, we can support HGMD.
Okay, let's see. In the log, do you use the operating system login ID, or is there a user login to the software?
That's a very good question. The software is licensed under a user model, so we are using that login. And in fact, you may have an environment where multiple users are sharing a physical machine, let's say in a lab environment, etc. So if you log out, and in that context, we actually have a flag to allow the software to be logged out whenever you shut it down. So starting up the software as a new user, you would get this login screen. And as you go through the login, come back and your information, your name is captured there, and then that login ID is what's captured in the log.
Thank you. I see a couple of questions regarding compatibility with different operating systems. Could you just briefly touch on that?
Right. So VarSeq is able to be run. It's a desktop tool. We don't require. We're not. Well, one important thing that's also I should have mentioned is obviously very relevant in the lab context is we're not sending any of your variant data that's coming into the software outside of your network. And in fact, the only thing we're using the network for is to download our public annotations and to log into our licensed system, etc. So it's a desktop tool. It runs on Mac, it runs on Windows, and it runs on Linux. We try to be as flexible as possible in those support configurations. We actually have a 32 and 64-bit version on Windows, and we'll run on the latest Red Hat, CentOS, and Ubuntu distributions just fine. So very flexible. It's actually a very high performance and very memory efficient. So even running these very large projects like this YIR Trio and going up to exomes, you can do this on machines with very moderate number of cores and RAM. You can see I'm actually only using, whoops, lost it there, VarSeq, about 250 megabytes of RAM just to work with this data. And it scales up. If you're working with whole genomes, you might be going up to a gigabyte, etc. But it can be worked on pretty much any analysis workstation. And in terms of, I think, in terms of how we do that, we don't write this in Java. We're writing this with high-performance C++ with cross-platform libraries to support all this great GUI information.
Okay, I see another question that just came in here. Do you plan to support other HGMD subscription models without having the download license?
So currently, that hasn't been our plan, simply because it's not economical for us to, because basically HGMD doesn't allow access to their database outside of the download context as a independent software vendor like we are, without going into a pretty serious partnership relationship like Alamut has with HGMD. We're definitely exploring that, and based on user feedback, we'll be pursuing that option.
Alrighty, thank you, Gabe. That's all the questions I see. Do you have anything that you want to add, or shall we wrap it up?
Sounds good to me. Feel free to follow up with any more questions via email, and as always, we'll be recording this webcast and making it available if you wanted to go through in more detail or share it with a friend.
Great, thank you so much, Gabe. We appreciate your time today as always. And thanks everyone out there for joining us, and we will see you next time. Enjoy the remainder of your week.
All right, thank you.